In this tutorial, we'll teach you how to use the Pathway and Annotation visualization tool called PANDA. The first thing that we do is we open up a web browser and we navigate to the bioinformaticstools.mayo.edu page. On this page, you'll find a number of software applications that we've developed in the Bioinformatics core at Mayo Clinic. Let's scroll down to PANDA. Here we can see that PANDA has a description as well as a place for users to sign up for updates. So as new releases come out, you can get information sent to your email. If you want to, that's not required. As we go down a little bit further, we have more information. Here we have the link that is to the tool itself, the GitHub repository if you wanted to download the code, supplemental files, which we'll use during this tutorial, and for more advanced users that may want to use custom pathways, we have another video that's created. So let's open up the page to the tool itself. As you can see, the first thing you need to do is log in. If you don't have a login, which I'm assuming that many of you don't, we'll go through the sign-up process. To create a new account, all you have to do is fill out these few pieces of information. And that's it. I've very easily signed up for an account, and I'm already logged into the toolkit itself. So this is the main landing page of PANDA. As you can see, it's broken up into a couple of different pieces. On the top is the toolbar, where there's different types of applications that you can select. Over on the left are the different annotation sets. So we have preset annotations here from DGIDB, OMIM, Human Phenotype Ontology, or HPO, Malacards, PharmGKB. And as we walk through the program, we'll upload our own annotation sets, which will appear over here. The bulk of it is this main table. This main table contains a link, which will open up the pathway, which we'll show in one moment, the source from which that pathway was derived, and the number of genes in that pathway. And what this allows you to do is to sort by different numbers. So cytokine receptor interaction has the most number of genes, and it's a KEGG pathway. So now let's make this a bit more interesting, and let's play with some real data. So let's go back to our home page, and we'll download the supplemental files that we mentioned earlier. It's very small, so we can upload this in no time. So here you can see that there are a couple of different files in here. One's a text document, a JPEG, a TSV, and a GIF. GIFs and JPEGs are both image file formats. So let me open this one up. You can see that this is actually what the JPEG looks like. And so I can know that that particular image is associated with some type of annotation as we walk through the tool. So let's look at this TSV file. Mine is set up to where it reads in Excel. And what you can see here is this is just a list of genes. It doesn't really matter what they are. Obviously, you would have one that is important for your experimental design. What I'm going to do is I'm actually going to create the input file, and this is all I have to do. In that first cell, I have to say that it's hashtag gene, and that's all I need. Optionally, I can put in other descriptions. So HRAS, this is my favorite gene, and I don't like this one. I mean, it doesn't really matter what it is. You could have any number of columns, any different types of annotation, and that's what makes PANDA unique, is this information will get brought back. So let's save that. We'll save it on our desktop. And we'll save this as something that we can understand as PANDA input. Doesn't really matter what the name is. But what is important is that we make sure that it's being saved as a text tab delimited and not an Excel format. So there we go. That is the input file. So we go back to our app, and let's upload this annotation. Let's go to our desktop. Load Panda input. So as you can see, it's uploaded the file, and we can give it another name here, or we could keep the original test input. We'll change it here. And this allows you to verify that the file format is correct. So it's specifically looking for the gene symbols and making sure that you're signing off saying that HRAS is the gene symbol. And it will also preview the first couple of lines so you can see if you have any other descriptors. So in this case, this is my favorite gene. This is shown. Now I've got to assign an icon to this particular set of annotations. So let's say this were mutated genes in patient X's tumor. Well, I want to assign this little life preserver to mean that's what this is. And we can upload many different files that are a little bit more intuitive or in terms of images. But for right now, I think this is a good starting point. So then all we got to do is finalize. And what it's doing now is it's taking that input file that we created just a moment ago, and it's inserting it into the Panda application. So if any of those gene symbols were invalid, they would show up in this text file, but of course none of them did because I had already checked it. But that tells you if you've tried to upload a gene symbol that we weren't able to find. And now you can see that we've added another column of information, and it's got that life preserver for this test input because that's the name that I gave it. And now I can sort it based on the number of variants or the number of entries, number of genes that are in this test input file. And we see that renal cell carcinoma, pathways, and cancer shows up. Well, that's great. I can also test to see if this is significantly enriched given the number of inputs and outputs by doing this enrichment test, which is just a Fisher's exact test. And so I can give it my another name, so Fisher test. And the annotation, I only have one tool or one annotation set that's uploaded, so I'll just go ahead and calculate. Now you can see the results of my Fisher's exact test, and I can sort these from high to low or low to high. And there we can see that we have a very significant Fisher's test score in renal cell carcinoma. So now I have the results of my Fisher's exact test, and these are my scores, as well as all the other information about the different pathways. Now let's actually explore what the data underneath looks like. Let's say I'm scrolling through these different pathways and I see acute myeloid leukemia. That may be of interest. It has a significant Fisher's exact test score, and I know something about it. So what I can do is I can open that up in a new tab, and now you're actually seeing the heart of what Panda does. So if you remember, I used this little life preserver to represent the test input data. And just like as you're looking at a regular KEGG pathway database, you can see that the genes for which my gene list actually contains them are shown with that little life preserver. So here is NRAS, and if we hover over that text, we can see that, okay, so there's the gene name and description. But I also have HRAS here, and that one had that description that it's my favorite gene. If I were to hover over each of these different genes, you would see that they have another gene symbol. And if I wanted to learn a little bit more about what PIK3R5 is, I could just click that and it takes me to gene cards. This is a very nice resource. And what I can do is I can very quickly look over the entire pathway, and I can see that this part of the pathway is much more enriched for that gene list than, say, this part of the pathway. So I could concentrate my efforts here. Alternatively, we could introduce some of our preset annotations, like drugability, OMIM, human phenotype ontology, mallocards, and pharmgkb. So as I toggle this on and off, I don't really see any pharmgkb genes. There are a lot of mallocards in these regions, and so if I were to click on one of those, what it would do is it would take me to either gene cards, or I could look at the relationship between this bad gene and pancreatic cancer from mallocards. And here I can read a little bit more information. Also HPO, a lot of these have human phenotype ontology terms, and we'll just look at examples of those. So some of these have actually a lot of them. So FLT3, they have abnormality of the blood and blood-forming tissues, etc., etc. DGIDB also has a lot of information. So looking at STAT3, if I hover over that, and let me just click on it, you can see there are several different relationships that come from different sources taken out of the DGIDB, and here I'll actually click the link to take me to DGIDB to make sure I get the most up-to-date results, and you can see that there's all sorts of information about drugs and the relationship to this particular gene. So that's just kind of handy to help you sort through and find relationships that may or may not be apparent. But as you can see, as I get too many of these guys in here, it becomes very difficult to sort out. And so that's really where you can take advantage of this toggling feature to just add or remove the different annotations you want to see or don't want to see. And that helps clarify the visual field. So now let's say I want to share this test data input with another user that's on the system. And I happen to know who that user is because it's me. So what I'll do is I'll go to data sharing, and here I haven't set up any groups, so I'll have to start by setting up a group information. So I'll call this my test group, and this is for demo purposes. And what it does is it says, all right, so these are the annotations that you've uploaded. Check which ones you want to share. So this is the only one I have, so I'm only going to check this one. And I know there's another user on here whose name is M087494. So I'll just create this group. And now you can see that the group was successfully created, and the group members are me, which I just did this. This is my username right now, and then my other username for a different project that I have. So as I log into this particular user, now this test input is available to me. And so that makes it very nice to share information with colleagues. So now that I'm done with this test input data set and the Fisher's test, I'm going to go ahead and clear these out and remove them from the database. And to do that, you just click the X. And the same for the test input. So now we're back to where we started with a blank slate, and hopefully this tool has helped you integrate multiple data sets such as genomics, transcriptomics, methylome data, drugability data. All of the information that you can imagine tied into the gene level can be accomplished through this tool. So we hope that you like it, and we welcome any feedback. Thank you very much.